So you all saw this incredible amount of cultures that we are gathering through this Dimensions of Biodiversity project. So we are at 52,000 cultures that we're trying to integrate into the phylogenetic tree of life. There is an additional challenge here because we have also a lot of short reads with next generation sequencing of amplicon sequencing. And so we want to integrate all this into phylogenies, and as you know, the reason is because out of these phylogenies we can have those classifications that we need so badly. So that represents a huge challenge. So we're going, how do we integrate all this data into the description of new species? And not to criticize, because I am a systematist, but we've been slow. And there's good reason to be slow because we want to be careful. But at the same time, based on these numbers, we're talking about millions of species that are out there. When will we get to be, I think we can be more efficient. But the problem with this is that, in a way, we have this wonderful resources. Now we're having a lot of cultures, we have a lot of sequences, but also we have an increasing amount and diversity of molecular data. So we have, of course, Sanger sequencing of ITS and LSU, and we have Sanger sequencing of multiple loci. Now we are into next generation sequencing of genomes, transcriptomes. We have dd-rad-seq. We have next-gen sequencing of metagenomes and metatranscriptomes. And also, we have NGS, or a next-generation sequencing of amplicon sequencing. So all of this is really great. It's wonderful. And some of this is based, as I mentioned earlier, derived from cultures. But in some cases, there is no cultures. There's just sequences coming. Yet, we want this information. And the problem in this is that there's not that much more taxonomists out there to do the important piece of this, which is to describe all these new species. So there's this huge bottleneck. And I think to help, what we need is really these bioinformatic tools, powerful bioinformatic tools that can empower us as taxonomists and systematists to kind of open this kind of bottleneck that is there. Okay? So this is what I will be talking about, how to empower us systematists in doing this. So for this project, what we did was to, we sequenced the ITS LSU as one amplicon, and this is from ITS1F or ITS5 up to LR3. So that's a 1.2 kb. If you're going for ITS, why not, it's the same price, get a 1.2 kb, and you get the LSU that's alignable across fungi. And then you have the 5.8S also, not much variation there, but across all fungi, that's quite useful too. And then at the same time, you have spacer 1 and spacer 2 of the ITS to help you at the finer resolution level. Why don't we get all that together? And you still have the barcode, if you like, the DNA barcode, you still have it. But on top of that, you have LSU that can help you put those sequences into a phylogenetic framework rather than being stuck using only similarity with BLAST. Okay? So, and then from this, we exclude all the redundant sequences, so the identical sequences of this amplicon, and then we keep, though, the geographical and the ecological data associated with all these strains, okay, even if they're identical. And then we align the 5.8S and the LSU, and including all the AFTAL and GenBank data, then we do these very large phylogenetic analyses, and also involving also similarity. So now the goal here is to see where that sequence, that ITS LSU sequence from a group of strains, potentially in our case, where does it fall in the tree of life, okay? And we use both similarity and phylogenetics for this. And then the goal is that once you see where it falls, that's the beauty of phylogenetics, is we can keep track through time of what we have collected before, where it fell in the tree, and now we can see, oh, these new samples coming from far east of Russia, they're falling in that clade, and that we have sequenced before multiple loci. We don't need to sequence this one. But that one is forming a new clade that might be a new species. We never sequence that. Then we take one representative out of that, and we do we sequence all these loci that were selected from AFTOL1. So that's the large subunit, small subunit, RPB1, RPB2, mitochondrial small. So now with that, we do a second phylogenetic analysis. Okay, so we now have a multi-locus phylogeny with less taxa. Okay, and then with this, we now also, because now we have genomic data and we have also transcriptomic data coming, so we can all assemble that together. And then using this alignment cumulative supermatrix approach, then we can generate these large phylogenetic trees that are very comprehensive with multiple support values that come from all these separate analyses that are done through this cumulative supermatrix phylogenetic approach. Now, that is a result in itself because it's comprehensive, and also we can use this with stronger support value because it's based on more genes. Then we can generate a constraint, a backbone constraint tree, which is really, really important because we're asking a lot when we do this first phylogenetic analysis based only on ITS, which is basically 5.8S and LSU. We have hundreds and hundreds of taxa, and we're asking for that little stretch of DNA to resolve all this, and the problem is huge, as you know. So therefore, this backbone constraint coming from the multi-locus analysis is really helpful to kind of help in resolving that tree. Okay, so how does it work? Let's see. So this is an example of a cumulative supermatrix. So you would have all these taxa here. This is just an example that I created in PowerPoint. And then you would say these three taxa, we have genomes. These other three taxa in pale blue, we have transcriptome. Here, these are dd-rad6 data from 6 taxa. Here is the Sanger sequencing loci that we were targeting, but for some taxa we have only 2 of those loci, for other taxa we have 1, and you can imagine that that would be, for a given taxa, it might be this locus versus this one. So, and then we have at the very tail end here this next generation sequencing, amplicon sequencing, which would be just a short read for a given target. So how to integrate all this? So let's say that we now look at, let's say we do an analysis on the 6 species at the top here, and we get this tree here. And then what we can do is we can add in a second analysis these other 6 species here that were sequenced using dd-rad-seq. And then let's say that's where they fell in with the green lines. And it means now that this internode here has 2 support value, right? Because we have the support value from the first analysis, because we had this taxa and this one. And then, whereas this node, internode here, is there only because it occurred when we added this taxa that's shown in green, okay? And you can continue like this progressively adding taxa with different amount of data. And in the end here, this is just an example of 6 analysis, meaning that on this internode here, we would have 6 different bootstrap support value. Now, it's possible that at the 6th analysis, the support value is below 70%, and it could be below 50%. But the internode is still there. And all these other analyses perhaps were supporting it very strongly. So if you did only just put everything together, you get the result, you say, well, I cannot conclude anything because I don't have support. But if you see that all these other analyses were supporting it strongly, it's just by adding so many taxa that you lost the support, but the relationship is the same, perhaps we can have confidence in this result, okay? So this is one of the power of this method. And also what is really powerful is that, don't forget, we have information about that is associated with all this embedded in this tree. So for the endoclinic and endophytic fungi for which we have this information, we have phylogenetic information here now in this large comprehensive tree. We have also, we know what type of data. So for each of those strains, we know if it came from genomes, we have genomic data, or if it comes, we have only next generation amplicon sequencing. And also we know the amount of missing data. You know, if this is really something that we have only one small locus that we have, that's all we have, or if we have a full genome. And also we have voucher information for each of those strains, and we have geographic information and ecology. So that's really powerful. and it's all integrated into that tree. So this is an example that Betsy showed earlier, and here is just an example of when we integrate all this information into one tree. I won't talk much about this. It's just to show that there is structure in this in terms of geography. There is also structure, as you can see, in terms of the host. And what is also important is that we have alignments. And that's really the key point here. We have alignments for all these different loci here. And so, and we have also cultures, okay, for these endophytes. So let's say that you come now with a new bunch of cultures, let's say, or sequencing that you obtain, sequences you obtain, and they're unknown. You want to integrate them into this existing tree that you have. And let's say that you can, there is a method out there that could place them automatically into that tree for you. So you go online, you include your sequences into this kind of entry point on a portal, you click a button, and then it tells you, it shows where yours are falling. And imagine that, let's say they fall all into this clade. So let's imagine that you could click on that internode, and you could download all the alignments that are available, plus all the voucher information, okay? So, and then also you see, well, that's the sister group here. So if you click here, then you get also the data for the sister group for routing your in-group, okay? And you could click and choose whatever you want in this, you know, and then have the set that you want. Then you're ready to download the alignment, and it asks you, okay, do you want just the ITS? Do you want the large subunit? Which locus do you want? Or do you want them all? Okay, and you click and you download, okay? So that's what I'm mentioning here, downloading the alignment, and now also you could download the voucher table. So as a systematist and phylogeneticist, that would save so much time, because to me that's the largest thing. Assembling those big matrices is a lot of work, and also producing these very large tables, voucher tables, is an incredible amount of time, and we seem to be always starting back from scratch. There's always something, and we cannot really use completely what we had before, because now we have this and that, and it's really complicated. But if it's all integrated into databases that you can retrieve from a phylogenetic tree, that would be wonderful. Another way of doing this also is that you might say, well, often we do this, you will have your new sequences that are here in black, and you already went on GenBank and you blasted and you retrieved the most closely related sequences that are in there, and you built your own tree, or you use a MoldBlast, for instance, and you have your own tree. And now you could just compare and say, okay, is there any of the species in that tree that are the same in this big mega comprehensive tree? And you could say, well, yeah, it's all in this clade here, and therefore you know that if you click on that internode here, you would have, sorry about that, there you would have this, and you could download the alignment for this group. Okay, so you don't even need to place your sequences into this actual comprehensive tree. You could have your own tree and then compare with the tree that's available online and download these alignments. So, so I think, coming back to this problem, I think that with this approach, we would be able to open this kind of pipeline where there's this severe bottleneck, and then really have access to this data and obtain for us, a readily available alignment, readily available cultures, phylogeny for unknown fungi. Now, another thing, that's a dream though, because you could imagine also that you could order directly the cultures that are available. So you would see your group is falling in that group. You say, oh, okay, Arnold's lab have all these cultures that seems to be in the group that I'm working on. You click and you order. That would be incredible. That would be wonderful. But I think we're not far from this, because we have already the tools to download these alignments. So this is existing already. And we're testing it right now. It's working really well, actually. And that's what Ignacio will show you. And this is a new online service that we're providing as part of the endobiodiversity.org project, or the Dimensions of Biodiversity project. And this program, or this kind of process is called TBAS, for Tree-Based Alignment Selector. And that was developed by Ignacio Carbon, and he will give you the detail of how this works. Ignacio.